Ion exchange chromatography is one of the most popular methods of protein separation. In this method, the components of a mixture are separated based on their binding affinity towards an ion exchange resin. However, these resins often suffer from low salt tolerance. This makes the separation of proteins from biological samples, such as human plasma, which tend to have high salt concentrations, quite challenging, expensive, and resource-intensive, where the samples first need to be made suitable via dilution or filtration processes. In a recent breakthrough, scientists from Croatia, USA, and Germany have investigated a cation exchange chromatography resin that has much higher salt tolerance than existing commercial resins and found out it can separate clotting factor 9 from among vitamin K-dependent clotting factors in blood without any prior processing. To arrive at their results, the team conducted experiments and model simulations to evaluate two resins side by side, the sulfonic type and the sulfate type. The findings showed that the minor structural differences between the sulfonic and sulfate groups led to significant differences in salt tolerance and hydrophobicity. Further, the presence of the sulfate group increased the salt tolerance, affinity, and selectivity of the resin towards certain vitamin K-dependent clotting factors in the blood. This suggests that ion exchange chromatography resins with sulfate groups could be ideal for the separation of different components of human plasma-derived blood clotting factors. The findings from this study opened doors to implementing the recently developed salt-tolerant sulfate-based resin in industrial processes, which will allow optimization of the downstream processing of biologically active plasma proteins.